Hello everyone and welcome to the fourth lecture of mutation. In last lecture, if you remember, that is the third one, we discussed about the nitrogenous bases. Why? Because when we study mutation, it is a very important point to understand about the structure of nitrogenous bases or bases. So today we will discuss some more points that about the nitrogenous bases. As we all know that we have, if we talk about DNA, we have four kinds of bases. We have adenine, we have guanine, then we have cytosine and thymine. If we talk about the DNA, these two are the purines and these two are the pyrimidines. Already we have discussed that these two are the purines and these two are the pyrimidines. Since we all know that DNA that exists as a double-stranded structure or double helical structure that was given by Watson and Crick, and if we talk about these hydrogen bonds, the hydrogen bonds, these link together the two strands of DNA molecule. And where these hydrogen bonds are, actually these bonds are between these nitrogenous bases. So if I draw two strands of DNA, suppose this is strand number 1 and this is 2. This is how we draw a simple structure. We have not drawn the helical structure. Just for simplicity, we have drawn two strands that are straight. And in between, we have the hydrogen bonds. And we know that in between adenine and thymine, that means adenine, it always pairs with thymine. And in between, we have two bonds. And cytosine, it always pairs with guanine. And in between, we have three bonds. And this is a must pairing. That means always we will have two bonds, hydrogen bonds between adenine and thymine, and always we will have three bonds between cytosine and guanine. That means this way we can represent the picture of double-stranded DNA molecule. Now these are some important points. Next thing that is important to understand, that is about mutation at individual gene level. If you remember, when we were discussing about the changes or the types of changes in genotype, we discussed that the first type, it was change in chromosome number. Second was in structure, change in structure of chromosome. And third one, that is individual gene level. So whatever we are going to discuss, that will be about at the individual gene level. So first thing today we will discuss about, in short, what is a gene and how to define a gene. There are a number of definitions. There are a number of definitions of a gene. You can have gene. How to define a gene? That is sequence of nucleotides that code for a protein. And more accurately, if we study about the definition or structure of a gene, how do we define gene? In a simple way, if we understand the concept of gene, how to define a gene. Suppose here I have my DNA molecule, and if I say this is a gene, it will code for RNA, and ultimately this RNA, it will code for protein. That means on what basis, how we can define gene over here? That means it is a sequence of DNA. Sequence means we are talking about the sequence of nucleotide or base pairs. So I have a particular sequence over here that codes for RNA. Just remember one thing, how do we define gene? That it can be defined as sequence of DNA that codes for RNA. Now if this RNA or this gene is a structural gene or protein coding gene, then only this RNA will code for protein, otherwise RNA will be the end product. That means it is not necessary always that if I have a gene, always I will have the end product as protein. Sometimes, for example, RNA is the end product of a gene. For example, in case of tRNA, that is transfer RNA, or rRNA, that is ribosomal RNA. That means these RNAs, if they are coded by the gene, they do not code for any protein. That means this is the end product. So it is not always necessary that if I have a gene, always I will have the end product as protein. That means a gene is a sequence of DNA that codes for RNA, and this RNA may code for, it may code for protein or polypeptide. Now this is how we can define a gene. Now we will come to the next point, that is about molecular basis of mutation. When we study about the molecular basis of mutation, what does it mean? That means at molecular level we have to understand that what is the cause, where at molecular level the changes are occurring, and what molecule already we know that change in the DNA sequence. That means at molecular level where the change is occurring in a molecule of DNA. So this is what when we talk about molecular basis of mutation. In broad sense of broad classification, we can have two kinds of mutation. The first one, that is spontaneous, and next one we can have induced mutation. The first one, that is spontaneous, we will take up this first. Now, before moving on to spontaneous mutation, something very important to understand, that from where it has come, what happens during spontaneous mutation. In simple language, what is spontaneous mutation? It is simple mutation for which no known cause is there. That means it occurs spontaneously. And what is the cause? It might be like during a metabolic error, for example, error during DNA replication. That is one of the cause of spontaneous mutation. Now, if we talk about something about molecular basis, a few things are important. We all know that the double helical structure that was described by Watson and Crick. That was described by Watson and Crick. What they did, they described the double helical structure of DNA, and they also proposed what they proposed. Semi-conservative mode of replication of DNA. They proposed. On what basis they proposed? It was on the basis of base pairing, that adenine it pairs with thymine and cytosine it base pairs, or there is a pairing with guanine. So Watson and Crick, what they did, they described helical structure of DNA, and they also proposed semi-conservative mode of replication of DNA. And when they proposed these things, they also proposed mechanism of spontaneous mutation. Now, how Watson and Crick they proposed mechanism of spontaneous mutation? What they pointed out something in bases or nitrogenous bases. And what was the point? And they pointed out that if we talk about the structure of bases, that is not stationary or that is not constant. That means it can change. And what kind of change? That means what they pointed out, that structure of bases, it is not static. That means there are certain changes occurring in the structure of bases. So this is the point that they pointed out. And what kind of change? That change was shifting of hydrogen from one position to another. That means if I have a base, this is just a schematic representation. If I have a base, for example, if this is your base, what they pointed out, there is a shifting of hydrogen. That means if this is the hydrogen, it shifts from one position to another position. Now this fluctuation of this chemical changes is what we call it as tautomeric shift during the process of tautomerism. So this is the point that they pointed out, that it's all about the bases, and that's why we have studied about the structure of nitrogenous bases. Along with the structure, what Watson and Crick they pointed out about the mechanism of spontaneous mutation, that basis, the structure, it is not static. That means how this structure is changing due to the shifting of hydrogen from one position to another within this structure. Is it very clear? Now this is what they pointed out, and this change or fluctuation in the structure is termed as tautomeric shift. Now since we know that adenine, since we all know that adenine, it pairs with thymine, and cytosine, it always pairs with guanine. Now what is the effect of change in structure? Due to change in structure, what is the result of change in structure of bases? Base pairing is altered. That means under normal conditions, if I have a normal structure of any base, be it adenine, guanine, cytosine, thymine, whatever, suppose these are normal bases, it will always pair with T, and it will always pair with C, will always pair with G. This is under normal case. Now what will happen? If there is a change in structure of these bases, the base pairing is altered. That means now it is not necessary that it will pair with thymine. That means now it can pair with some other base. Now this is what Watson and Crick they pointed out about the spontaneous mutation, and because of this change in the base pairing pattern, or due to this change or this alteration in the base pairing of these bases, we have a different mutant, or we say that mutation occurs. That means if mutation, that means here we have changes in the base pairing of bases. Now this is what they pointed out. I think that is enough for today. In next lecture we will discuss what kind of change and what kind of changes occur in base pairing. So I hope whatever we have discussed, that is very clear to you. Please, if something is not clear, whatever we have discussed, go through the previous lecture where we have discussed about the structure of bases. In my next lecture, we will discuss about that what kind of change and what kind of mutation actually is occurring due to the change in the structure of bases. Whatever you feel like, please do comment in the commenting section, and thank you for watching this video.